Inside the fluid interior of a eukaryotic cell, past the studded membrane of the rough endoplasmic reticulum, sits a highly organized, multi-layered structure. Prokaryotic cells manage their secretions using a relatively simple translocation mechanism directly on their plasma membrane. They lack the dedicated infrastructure required to handle complex, multi-stage molecular processing. Eukaryotes rely entirely on the Golgi complex. It is the central organelle, the secretory pathway, operating as the routing hub for the entire cell. It receives newly synthesized raw proteins and lipids from the endoplasmic reticulum. It then chemically modifies these molecules, sorts them based on their final destinations, and dispatches them across the cell. Without this central processing hub to direct intracellular traffic, the targeted distribution of materials necessary to sustain complex multicellular life would be impossible. In 1898, while observing the nerve cells of barn owls and tats, Italian biologist Camillo Golgi identified a distinct structure he called the internal reticular apparatus. 16 years later, Santiago Ramon y Cajal formally termed them Golgi bodies. The precise architecture of the organelle remained elusive until 1954, when Dalton and Felix resolved its structure using the electron microscope. Early researchers struggled to define the Golgi because it is extremely pleomorphic. Its physical size and shape vary drastically depending on the physiological state of the cell. In human leukocytes, the Golgi forms a compact spheroidal mass near the nucleus. In neurons, it spreads out as a broad, extensive network across a large area of the cytoplasm. This physical variation perfectly reflects the specific secretory demands of the host cell. The architecture adapts to the exact volume and type of cargo it needs to process. The foundational unit of the Golgi apparatus is the cisterna, a single, flattened compartment. 5 to 8 of these bundled together in parallel, separated by a uniform space, forming a vertical stack called a dictyosome. To maintain these precise gaps without collapsing, the structure relies on an internal skeleton. A class of proteins known as grasps form rigid scaffolds between adjacent cisternae, locking the entire dictyosome into a highly organized stack. This diagram illustrates another class of matrix proteins called Golgins. These form long tethers, composed of stiff coiled domains that extend up to 400 nanometers from the Golgi surface to secure incoming transport vesicles. The result is a highly rigid, tethered framework primed to capture arriving cellular cargo. In mammalian cells, multiple dictyosome stacks are laterally interconnected by tubular membrane bridges. Together, they form a sprawling macrostructure known as the Golgi ribbon. The flow of material through this ribbon operates with strict directional polarity. This cross-section shows the receiving cis face, capturing vesicles from the endoplasmic reticulum. Cargo moves into the central medial cisternae for biochemical processing. Finally, it reaches the shipping transface, packaging finalized products into vesicles that bud into the cytoplasm. This physical polarity dictates a strict one-way assembly line for all cellular cargo, progressing directly from the cis receiving face to the trans shipping face. Inside the mammalian Golgi, there are more than 250 distinct enzymes, organized in a highly specific sequence across the separate compartments. A primary task of these enzymes is the extensive modification of N-linked oligosaccharides on the glycoproteins arriving from the ER. This diagram outlines the exact pathway. In the cis Golgi, red mannose residues are stripped away, and a purple N-acetylglucosamine is attached. In the medial cisternae, green fucus and orange galactose are added. Finally, a blue sialic acid completes the chain. By separating these enzymes into distinct physical compartments, the Golgi ensures that glycoproteins emerge with a staggering variety of precise modifications, perfectly customized for their specific physiological roles. Beyond processing proteins, the Golgi has a critical secondary mandate: lipid metabolism. While basic glycerol phospholipids and ceramide are synthesized in the endoplasmic reticulum, the Golgi takes that ceramide and converts it into complex structural lipids. This diagram shows that process. The Golgi converts ceramide into sphingomyelin by transferring a phosphorylcholine group. Alternatively, it yields a variety of glycolipids by adding one or more carbohydrate sugar residues to that same ceramide base. In plant cells, this metabolic capacity expands even further. The plant Golgi synthesizes complex branch-chain cell wall polysaccharides like hemicellulose and pectin. The synthesis of these cell wall materials is so demanding that up to 80% for regulated secretion, specific proteins enter secretory granules, awaiting an environmental trigger. Finally, the constitutive pathway provides direct transport of proteins and lipids for immediate incorporation into the plasma membrane. Without these precise tagging and routing protocols, lytic enzymes, hormones, and structural lipids would chaotically misfire across the cytoplasm, rapidly leading to cellular death. This leaves one final mechanical problem. How does a dividing cell split this massive interconnected ribbon structure equally between two new daughter cells? During the G2 phase and prophase, mitotic protein kinases phosphorylate the Golgi matrix proteins, forcing the internal scaffolds and tethers to release their grip. The entire ribbon unstacks, breaking apart and vesiculating into a dispersed Golgi haze of thousands of drifting fragments. By telophase, these tiny fragments are perfectly partitioned. Dephosphorylation allows them to instantly reassemble into two identical Golgi ribbons in the new daughter cells. From the intake of raw materials at the cisphase, through the highly compartmentalized modifications of the medial cisternae, to the molecularly targeted routing of the trans-Golgi network, the Golgi apparatus operates as the ultimate arbiter of eukaryotic order. It is a dynamic, self-assembling factory that ensures every manufactured molecule reaches its exact required destination.